Good afternoon, my name is Quinn Remick and I'll be presenting my group's gut microbiome project to you. Now obviously we're working with the gut microbiome, which is the portions and the types of microorganisms living within a given location. We're testing the relationship between the gut microbiomes and physiological conditions. The question that we were testing for is what essentially is the difference between the gut microbiomes of dogs. We were testing to see if there were similarities in the gut microbiomes of animals of the same species. Our hypothesis would be that there were a few differences, a few similarities, but most of it would be different. Our rationale is that there would be one or two or maybe a few different bacterium that were genetically linked to a species, but that these were very few, and most were affected by other environmental factors. Now, the methods that we use, cut this out. Now the methods that we used, first we used a QIAGEN kit to isolate DNA, but we did this by adding an inhibitor buffer that removed basically anything that would prevent us from isolating the DNA, and then we heated it, vortexed it, and centrifuged it. Then we added proteinase K to destroy proteins that degrade the DNA. We added ethanol, which allowed us to we then added ethanol, which brought the DNA into the spin column filter. And then we added a wash buffer and we centrifuged it. And then after that we added a elution buffer, which allows the DNA to just flow through the spin column. After that, we used a Zymo kit, which was used to concentrate the DNA. Most of this was accomplished by adding various buffers and centrifuging the DNA, which was finished with adding an elution buffer, and this eventually gave us a DNA concentration of 10 times what we originally had. We then wanted to check what the concentration of the DNA was, so we used UV spectrophotometry to see if the DNA was contaminated at all. What we were looking for was a 2060 to 2080 ratio, and so DNA absorbs at 260 nanometers and proteins absorbs at 280 nanometers. And a ratio below 1.5 would indicate that we have possible contamination by protein. As you can see, for I'm looking for 1.5. Okay. UV spectrophotometry. So here we have our UV spectrophotometry results. For 555, for sample 555, we got that's the total reads. A table. Oh, fuck.